We get one more minute of biology fun, huh? The one on the left was an actual statement by Donald Trump right when he became president, because he promised to like repeal and replace and all this stuff, Obamacare within like the first year of being president, and he actually said nobody knew healthcare could be so complicated. So I always think that's a good one. That man is our president. All right, finishing chapter 18, which is the final things that Dr. Bogorski covered on Friday, and then getting into chapter 25 and 27, which is what Dr. Mott talked about today. Dr. Mott is back. Who's excited? You guys miss him? I miss Dr. Mott. Not that I don't like Dr. Bogorski. He just comes back and I'm like, oh yeah, you teach this class too. All right, is that going to work for me? Just a reminder, I have not gotten very many responses for this collaborative final, practice final that I'm trying to do. Again, if I don't get enough questions sent from you guys, I'm just not going to do it, I'll be honest with you, because I don't have the time to individually make my own practice final when I have 5 of my own real finals to take. So if I get enough participation, if I get enough questions from you guys, hopefully I will, since there's 1,000 students in the class. Even if every one person in the class sent in one question, I could make like 100 practice finals, right? Well, not 100, 50, I don't know. I could make a lot of practice finals, right? So just everybody, even if everybody in this room sent in one, we'd probably have enough to make a pretty good practice final, right? Again, I don't expect you guys to do everything. I would probably put in at least 50% of the questions, but that means if you guys send in 20 questions, I'll put in my own 20 questions and we'll have a 40-question practice final, right? So you're going to want to do this, I promise. All right, your biology fact for the day. The lightest touch that a human can feel is a bee's wing falling on your face. I don't know who took the time to figure this out, but they found that the absolute lightest thing you could feel is if they took a bee's wing. I think they held it like 2 centimeters above your face and dropped it. The hairs on your face were able to pick that up. So in case you were wondering, what's the lightest thing you can feel? Study tip. First I have to explain the picture. I have a friend that every time he sends a Snapchat, it's that face, every time. And so we make a lot of fun of him for it. I have like 19 screenshots of that face. And I went and I was like, well, take 20 seconds to look somewhere else. So I'm like, I'll just Google a picture of staring into the distance. And then I was like, why? I have so many. They're all just him staring at every Snapchat. It's a good time. So anyway, the study tip is, for every 20 minutes that you study, take 20 seconds to look somewhere else. It sounds dumb, but it really is partly for your eyes, right? You get far less strain from staring at your screen. I would also recommend downloading a blue light filter. If you spend a lot of time in front of a screen, all that blue light coming out of your screen, which is more than you probably think, like a white screen has a lot of blue light. That's really bad for your eyes too. But also just it gives your brain a chance to just take a break and go, all right, I've been staring at the same spot for 20 minutes. Just look out the window, look at your friends or boyfriend or girlfriend or dog or mom or dad, or even if you have to get up and go somewhere real quick, just look at something else to give your eyes a break for a second. Don't look at your phone. That's the same problem, right? The idea is to look away from a screen. So don't say, yeah, I'm looking away from my computer because I'm looking at Facebook on my phone. No, it's not how it works. Plus, nobody uses Facebook anymore. All right, so 2 ways to create different cell types. They're up there on the board, and I could sit here and I could talk at you about them. Much better if you guys explain it to me. In fact, the best thing, we're going to pair up. Just find the person closest to you, get into groups, 2s or 3s, and one of you explain A, cytoplasmic determinants, and one of you explain B. You have 1 minute. Go. I'm serious. Group up. I will stare at you until you do it.
You're my partner.
Fair enough. Everybody, pair up. 1 do A, 1 do B.
All right. I have no idea.
Do you know either of them very well? Which one are you more confident in?
pro-induction.
So very rude of you. Okay. Now, one of you who did not explain A to your partner, explain A to me.
A is where you have the fertilization of an egg, and the egg is not, certain chemicals in the egg aren't diffused yet. They're not diffused around the egg, and so when the cell splits, it then has chemical A in one cell and chemical B in the other.
Perfect. And so if chemical A circles is for brain cells, then that cell will eventually become a brain cell. And if the triangles in the second cell are for a liver cell, then that cell is eventually going to become a liver cell. Spot on. Somebody who didn't explain B to their partner, explain B to me. We learned about this 4 days ago. You guys all go on a bender this weekend? Forgot everything?
Yeah.
Come on. I heard lots of talking, and I know you didn't just talk about A.
So we talked about how it's kind of like peer pressure is what he was saying, kind of leads them.
Yeah, it's like peer pressure. Elaborate for me. I don't know what peer pressure is.
So the cells nearby are telling it, oh, go and do this, so then it changes
Yeah. So these cells on that bottom row communicate with the cells of the upper row and peer pressure it into becoming the type of cell that's most convenient for the organism. If that's, a heart cell, if that's a skin cell, if that's a brain cell, it just says, hey, this is what I am, and this is what you should be. Okay? That's how you can get different cell types, right? Because we start as just two cells, a sperm and an egg, right? I mean, that's not exactly how it works, but you get what I'm saying. And so how do we go from a sperm and an egg to an entire human being? One of these two ways, right? It gets more complicated than that, obviously. We're a little more complex than two methods, but for this class, just be familiar with those two methods. Okay? Does everybody feel pretty comfortable with that? Less comfortable with B than A, apparently? Go home and study, what is it, induction by nearby cells? Okay? Watch the Panopto, read it in the book, Google it, whatever. Be confident in that, okay? This is something you could absolutely see on the test. Okay, genetic regulatory cascades. A lot of words up on the slide, and that's all fine and dandy. I want you guys to understand the words, but more than that, I want you to understand the concept, okay? What are the light switches? Why the heck light switches? How does that have to do with anything?
stuff on and off.
Okay, yeah. Turning what on and off in this case?
The cascade.
Cascade. What does the cascade do?
Genes.
Huh?
For genes.
For genes, yeah. So we're cascading our genes to do what? Huh?
Represent like expression.
To eventually, yeah, express a gene and get a phenotype, get a physical trait, right? So far left switch is titled master control gene. What's that?
When you have one of those genes, it causes a lot of other genes to react for a specific trait. Like we use the SRY gene as an example in class.
Yeah, perfect. So SRY is a classic example of a master control gene. What is SRY?
Determines if somebody's a male.
Yeah, it determines whether or not you become male, right? Because female in mammals is the default sex, right? So in lack of SRY, you become female. With SRY present, you then turn on a whole cascade, right? So that you develop testes instead of ovaries, more usually like body hair, deeper voice, broader shoulder, you know, whatever you want to consider the typical male characteristics, and obviously along with more like, you know, testosterone versus estrogen and all that, right? So SRY is the master control gene. You turn it on, you get all those. You don't turn it on, you get none of them, right? All or nothing. It's like a master switch, right? If those light switches in the back had a master switch and the switch was off, I could sit there and mess around with the other switches all I want. None of these lights are going to turn on. But if I turn all the lights up and then the master switch, they all turn on at once, right? So all of these switches are ready to go. They're just waiting for the master switch to come on, okay? Is everybody feeling that? Is anybody confused on that? Have questions with that? I see some confused faces. Please ask.
What's another example besides the SRY gene?
Like another real-world example
Yeah.
of a master control gene? I can't think of any off the top of my head. Does anybody know any?
MyOD.
MyOD, yeah, that was one of them. Do you guys remember MyOD? I actually have some slides on it in a little bit here. Do you remember MyOD?
Yeah, it says here, semester triggers precursor to cells to differentiate into muscle.
Yeah. So you have your genes sitting there, right? And our genes are the same throughout our entire body. But then MyOD, the master regulator, comes in and says, you should become a muscle cell. And then all of the genes that turn an undifferentiated cell into a muscle cell, right? And it builds its actin and its myosin and its sarcolemma, all that stuff is just because the MyOD came in. Does that make sense? Do you see how it's parallel to SRY? That is a perfect example. I couldn't think of any off the top of my head. Does anybody know any others? I can't really. I'm terrible at like just thinking of them off the top of my head. And I'm sure if you Googled, you know, master control gene example, you could find lots of them. Other questions? Okay. Oh, look at that. Here it is. So MyOD is your master regulatory gene. You have an embryonic precursor cell. What's that? Undetermined cell. Undetermined, undifferentiated. It's just technically nothing at this point, right? Doesn't do anything. It's just there. Now we have a myoblast. Visually, structurally, how is it different from an embryonic precursor cell? It's the same, yeah. Do they look the same, right? It's because they are. A myoblast is determined, but it is not yet differentiated. And we'll go over that vocab in a second here if you're like, what? So it is going to become a muscle cell, but it is not yet a muscle cell. Why not? What's missing? I heard something.
I don't remember right. Nucleus?
It has a nucleus.
Oh.
Has a nucleus.
That cascade hasn't been started yet?
Yeah, and what starts the cascade? Master regulator, in this case, MyOD, right? So it just doesn't have that MyOD what's the word I'm looking for, attached to its mRNA, right? So when it actually becomes a muscle cell, a fully differentiated cell, right, it is a muscle cell, and it is not anything else. It's because your MyOD attaches to that region of the mRNA that codes for it, and then that starts that whole cascade effect, right, where you start getting everything, actin, myosin, becomes a sarcomere, all that, right? Cascades down, okay? It's just another example of how master regulators work. But remember, if you are given a specific example of something in class, of a process, of a cycle, of a type of whatever, if you're given a specific example in class, be familiar with it. Because you could see something as simple as, what is MyOD? And you would have to know a master regulator that helps differentiate cells into muscle cells. Fairly simple, it's basically just a vocab. Or you could get a question as far as, if you have a determined myoblast but a defect in your MyOD, what will occur? And you would have to know it will never differentiate into a muscle cell because the MyOD is not present. It can be determined all at once, but no MyOD, no muscle cell. Okay? So specific examples, pay attention. Okay? Questions on this? Okay. So, cells are determined before they differentiate, right? There's an order. Does anybody remember? There's 3 pieces. 2 of them are up there. I'll wait.
I can tell you 2.
Which 2?
Precursor and determined.
Ooh, close. You have one of them. Determined.
So close.
Determined. Anybody see another one that ends in ED? Differentiated. Okay. The third one is very similar to differentiated. It is undifferentiated. It's been 4 days, guys. Come on, come on. Okay. So there's 3 stages: undifferentiated, determined, differentiated. Next slide goes over the definitions. We'll get to that. But you have to be determined before you differentiate, right? There's no skipping of steps. Has to be undifferentiated, then determined, then differentiated. Always. Always in that order. Okay? It's also irreversible. That is something that's important to note. Okay? This is actually what makes stem cells so valuable. Have you guys heard of stem cell research? And there's super crazy stuff that they can do with it, right? I remember I came downstairs one time and my roommates were watching Family Guy, right? Don't really watch Family Guy all that much, but the main character had something wrong with him. He'd been like blasted by some radiation and was like all kinds of messed up, right? And he walks into a stem cell research lab and comes out 15 minutes later and he's fine. And he's like, why aren't we researching this? Why aren't we funding this? There isn't a lot of funding for stem cell research because of the ethical reasons. You get most of your stem cells from embryos, human embryos. So it's kind of like, you basically like steal them from pre-babies. So it's like, is that okay? I don't know. Anyway, that's what makes stem cells so valuable. Stem cells are undetermined, right? Once I have a muscle cell, it cannot go back to a stem cell. So I can't take a muscle cell and say, I actually have too many muscle cells, obviously, and I want that to be a brain cell, right? I obviously also need more brain cells. You can't. Okay, it's a muscle cell till it's dead. And you can't like recycle it and turn it back in, right? Nothing. It is until it's dead. And then it's dead and it's gone. Okay? So stem cells are valuable in the sense that they're undetermined. So you could implant a stem cell anywhere, theoretically, and it would become whatever it needs to become, right? So it could cure all kinds of diseases, right? Diabetes, right? Your pancreas is dead. Inject some stem cells in there, basically grow a new pancreas, right? Pretty crazy stuff. Anyway, that's my side tangent on stem cell research. I think it's super cool. So, the vocab. Undifferentiated, determined, differentiated, right? And that's the order that they come in. Spitball at me some just layman's term definition of undifferentiated, in your own words.
No purpose cell.
No purpose cell, okay, so that's one way of thinking about it. It's not anything. It's not going to do anything.
Is it a stem cell at that point?
Stem cells are essentially an undifferentiated cell.
Can it be kind of like all-purpose because it's not specialized yet?
Yeah, you could also think of it as an all-purpose cell, right? Eventually, be careful, it's not that it can do everything, but it could become anything.
Yes.
Right. So it could become anything. Like all-purpose flour could become a cake, could become a brownie, could become a cookie, could become the breading on fried chicken, right? I know so many uses for flour. I know 4. Okay, so it's not on any path to become a certain cell type. It can become anything, right? So I compare this to the undeclared college student, right? You are not a pre-med, pre-dental, pre-law. You are not a chemistry major. You are not an econ major, right? You are undeclared, and there's nothing wrong with that, right? At some point you should, but nothing wrong with even just, your first couple semesters being undeclared, because you can become anything, right? This is my motivational speech. You can do whatever you want to do. You can do whatever you want to put your mind to, right? Except go to space. We don't do that anymore. Russia does that, so you can pair with Russia. Is everybody feeling that one? We good on that definition? Okay. Determined, then. I was about to give it away. Layman's definitions. Come on, just throw them at me.
A declared college student.
It's a declared college student. You stole it. It's actually, I think, word for word what I have up there. Are you looking at my slides? I already put these up, so you could cheat. Come on, other ones. Yes, it's like a declared college student. Why? And remember what determined means.
It knows what it wants to be, but it's not there yet.
Yeah, so I am pre-med, right? My major is human biology, right? Am I a doctor yet? No, not quite. Super close, almost there, 10 more years. No, but I'm on my way, right? Fingers crossed. Someday, I will finish this path and become a doctor, right? Same thing pre-law, right? Is a pre-law student a lawyer? No, they like to pretend that they are, and they all think that they are. They are not, right? They have not passed the bar. They are not technically an attorney. They cannot practice, but they're on their way, okay? Same thing with econ majors. None of them are billionaire, genius, playboy philanthropists, but they all think that they are. All right, so declared college major, right? Going to become something on a definite path, but not technically that thing yet. So cells, right? It is determined to become a brain cell, but it is not technically yet a brain cell. It's on its way. All right, third one then, differentiated. Throw them at me.
Somebody who is, like, for when you're a doctor.
Yeah. I'm a doctor. I am differentiated from a mechanic, from an attorney, from an actor, right? I'm separate. I'm a doctor. What else? Take it more biology terms. What's a determined cell?
It's a brain cell.
It is a brain cell, right? And you can specifically look at it and say, this is not a heart cell, this is not a stomach cell, this is not a white blood cell, this is a brain cell, right? It is different from everything else, okay? So, it is a graduate. It has begun working in its field. It is doing its job as a heart cell, right? You guys see that? Are these comparisons working for you? Do you have questions? I see a hand coming up. Tentative question.
So, does every cell have to wait for a master regulator to become a differentiated cell?
I don't know that it's every cell. I know that's the most common pathway, and that's the one we focus on in this class. There may be other ways. Not really something that we concern ourselves with. Again, I always be careful with saying never and always. There's probably an exception, but we focus on the master regulator pathway. It's the majority of them. Other questions? Excellent. Let's talk about proto-oncogenes. Definitely understand all of the mechanics behind cancer, okay? That is something that Dr. Pogorski is very passionate about. It's something he's done research in. So first of all, he knows everything about it, right? Second of all, he cares about it, so you should at least care enough about it to study it for the test, okay? Figure that out in all your classes. Figure out what does your professor care the most about. I'll give you guys a hint. Dr. Mott, plants. Freaking loves plants, okay? It's what he does. He's a botanist, right? He studies stomata on plant leaves. So you're going to get like 10 questions just about stomata, okay? Because that's what he does, like 9 to 5, Monday through Friday, right? That's his other job besides teaching this class. So if you have a professor and you know, oh man, they love substitution reactions in chemistry, that's like what they study, that's what they do their research in outside of teaching this class. So you can bet you're going to get more questions about it. It may not seem fair, but whatever the professor likes the most usually is what you see tested more. So proto-oncogenes related to cancer, study the crap out of them, okay? Going to see that on the test. So they're a normal cell, okay? They promote cell growth. Proto-oncogenes, normal, healthy cell growth promoters, okay? They are just what makes your cells get bigger and multiply. There are tons of them. You don't have to know any of them. I don't know any of them. But the issue becomes when it suffers a mutation, right? And it stops working how it's supposed to. It works, I compare it to a stuck accelerator, okay? Because it's a growth factor, right? So proto-oncogenes tell your cell to grow. You get a mutation and your proto-oncogenes say, go nuts, grow as much as you want. And it's like when you put your foot down on the pedal and you just hold it there, right? And your car just goes. And you can't let go, right? There's no stopping it. It's like a brick on the accelerator, nonstop, okay? So that's part of how you get cancer, right? A tumor. Any of you have seen tumors before, you know they're pretty sizable growths that like oftentimes will stick out of your skin, okay? Because the cells that have the mutated proto-oncogenes that are now just called oncogenes, right? If it's an oncogene, that's the cancer-causing one. Proto, good. No proto, bad. They'll just grow and they'll push and make huge tumors, okay? All cancers, okay, have some kind of oncogene, some kind of mutation causing an oncogene, better said, okay? So if that's also another motivator, if you want to go in to study cancer or anything related to it, you've got to know proto-oncogenes and oncogenes, right? So how do they happen? 3 different ways. Okay, pair up again. You can do it with the same people you did before. One of you explain this one. One of you explain this one. And then the first one explain that one again. And the next one explain that one again. Okay? 2 minutes, go. Pair up, explain how they happen. Look at your notes if you have to. Just best guess off of the slides if you have to. Admit that you have no idea if you have to, and maybe your partner is nice enough to explain it to you. All right, it's gotten relatively quiet, so I'm going to take that to mean most of you are done. If you're not, that's okay. I don't want to spend any more time on this than we have to. So, who feels fairly confident about that far left one there, translocation or transposition, and can explain it to me? Because I don't know. I don't know how it works.
The one on the far left is where the proto-oncogene goes into the DNA sequence, and it's there in a really hyperactive area, and it just keeps getting hit time and time again, and creates a lot of growth stimulating protein.
Yeah, so our promoter, I don't know why this pen, my goodness. You guys know what I'm trying to circle. The new promoter over there on the far left, it got moved, right? And for whatever reason, you don't really need to know the specifics of it, but essentially that new promoter got put into an area where it makes the oncogene, the proto-oncogene, which is now an oncogene because of the mutation, work faster than it's supposed to. So if you were supposed to get one of those little bluish, purplish orbs down there, you got 3, right? I know that's not how it works. I hope you know that's not how it works, but it's a visual comparison, right? You get 3 times as much as you're supposed to in that specific case, right? So the promoter got moved, it makes the oncogene too active, you get too much protein, you get too much growth, okay? Who wants to help me with the next one? Gene amplification. Very similar idea.
So you can just get extra proto-oncogenes, and they're just producing the normal amount, but there's too many of them, so you get too much growth stimulating protein.
Yeah, that's perfect, spot on. You used to have 1 in this case, let's say, proto-oncogene there. Now you have 3, so now you get 3 times as much as you should have, and your cells grow too fast, okay? Again, 3 times is not a rule, that just is how it happens in this drawing, right? It's just that there's too many proto-oncogenes, you get too much protein. Help me with the next one, point mutation within a control element.
Is the control element basically just telling the gene to continue producing past when it really should?
Yeah, so here's our control element. I don't know if the pen's really going to help me. So we have our control element that is controlling the proto-oncogene, right? Some kind of mutation happens where now the control element, I guess you could think of it as like talks too much to the proto-oncogene, and it tells it make more, make more, make more, make more, make more, when it's really not supposed to be making that much. So the control element's what's actually out of whack, right? It's like you turned up the volume too high. And then help me with the 4th one, point mutation within the gene. This one's different. Obviously, you can tell from the product.
You don't get like extra proteins, you just get proteins that don't die.
Yeah,
Pretty much.
your proteins are too strong, they're too active, so some kind of point mutation within your gene made a super protein, right? An indestructible Hulk protein. And so normally some cell would come by and say, all right, chill out, you've grown enough. And this cell's like, no, busting through the walls like Kool-Aid Man. Doesn't care, right? It just does whatever it wants, nobody can stop it. It's a Hulk protein. That's how I always thought of it. Questions? You guys see how it works like the Kool-Aid Man? Me either. Real questions.
So do all these lead to cancers because there's too much of a normal cell? Isn't it cancer normally like an altered cell that does things it's not supposed to, like with the protein?
We're getting there, but so this is only part of it. Part of the cancer is excess protein leads to excess growth. And there's 4 different ways that that can happen, but essentially too much protein or too strong of a protein, too much growth, part of the equation for cancer, right? Protooncogenes alone, well, I guess oncogenes alone will not cause cancer, right? You could have tons of oncogenes. Does anybody remember why that alone doesn't cause cancer?
Isn't it because you have to have tumor suppressor cells not working?
Yeah. So the tumor suppressor cell, on the other hand, says slow it down, right? So you could have oncogenes and they could be going crazy, but then the tumor suppressor gene is like mom and still comes in and says, chill out, You're way too much, you're way too hyper, calm it down, okay? It's when you have a mutation in both, right? Things are going out of control and there's no mom to come in and say, chill out. Then it's nonstop, right? So tumor suppressor genes, it's like when brakes stop working, okay? So you can have the issue of a stuck accelerator, but I guess that could be solved by also hitting the brakes, right? Very bad for your car. But even if your accelerator gets stuck, you can still press on the brakes and slow down. Your engine will go nuts, but you can slow down, right? If you get a mutation in your tumor suppressor genes, they stop working, no one's there to say stop. So it's like you cut the brake lines, right? You can pump on the brake all you want, nothing's going to happen, okay? So it's just a decrease in the activity of the protein that enables tumor suppressor genes. So you have to have both to get cancer, right? So these are also seen in all cancers. Confusion on tumor suppressor genes? Questions? Comments? Okay, so kind of an idea of how, this is a very simplified form, right? Normally you have your protein kinases that interact with p53, which is just a type of tumor suppressor, and it gets transcribed and it says stop when it needs to, right? It says, hey, you're growing too fast, calm it down, right? If you get a mutation, there's a change in shape of your p53. It will no longer interact with those kinases, and it will no longer tell your cell to stop growing, right? So both of these cells had UV light, and both of these cells ended up with damage. What's the difference?
The suppressor just doesn't work.
Okay, and why doesn't the suppressor work?
It's like mutated.
Yeah, the mutation changed the shape of the p53, of the tumor suppressor. This mutation got fixed, or perhaps was a silent mutation or whatever, and so p53 is unaffected and still does its job, right? Because mutations get fixed all the time. You have mutations nonstop. It's actually pretty normal. UV light, we get that all the time. Standard. If it doesn't get fixed, that's when you have a problem. Cool beans? Yeah. So again, you have to have both. You have to have overexpression of protein, growth protein from an oncogene, and a lack of tumor suppressor gene protein. You get increased cell division, i.e. tumors, i.e. cancer, right? You guys see the relationship there? So you have to have both. I would not be surprised if some kind of test question came up about needing both of those for cancer. Okay. Let's get on to Dr. Mott's material. We have about 10 minutes to talk about all of life from the last 4 billion years. So, this portion, at least, is pretty much just memorization of dates. And I feel bad that that's what it is, but that's what it is. Life originated 3.5 to 4 billion years ago. Okay? That's date number 1 to memorize. 3.5 to 4 billion years ago, life originated on this planet. I made you guys a handy-dandy little list. I hope you are greatly appreciative of what I've done. Take a picture, write it down. Also, I posted these slides, so just you can also download it and look at it later. But these are the important dates to memorize, okay? First cells, 3.7 billion years ago. Stromatolites, type of fossil, 3.5 billion. Oxygen-producing photosynthesis, 2.7 billion. First eukaryotes, 2 billion, right? And on and on and on. But there is a logical order to these events, right? The first set, you had to get cells before you get fossils, right? Because fossils are made of cells, okay? You had to get photosynthesis before you got eukaryotes. Why?
Chloroplasts are prokaryotes.
Chloroplasts are a prokaryote in eukaryotes. What else? What do eukaryotes breathe? I thought you raised your hand. Oxygen, right? So we had to have photosynthesis to make oxygen for oxygen-breathing organisms, right? Then you get multicellular eukaryotes, right? Because you're not going to just, it's a walk before you run type thing, right? You get a small eukaryote before you get multicellular eukaryotes, right? Then you get marine animals, right? Because marine animals are multicellular eukaryotes, okay? And the atmosphere was not excellent for surface dwelling at the time. Then you get land plants and animals, right? As the atmosphere chilled out and the gases got more breathable, more survivable, then stuff started moving on to land. Then you get humans, because we obviously are land animals, 200,000 years ago. Okay? So I would just, as terrible as it sounds, you got to memorize all those dates and just understand the logical sequence, why it had to happen in that order. Mostly, I would say the most important one, the most emphasis, is why we had to have oxygen-producing photosynthesis before eukaryotes, right? And again, it's just eukaryotes need oxygen to breathe, therefore photosynthesis had to occur before we could get eukaryotes. Okay? Cool beans? All right. Who's ever heard of Louis Pasteur before this class? Most of you, right? If you haven't, you really have. How many of you have heard of pasteurized milk or pasteurization? It's named for Pasteur, right? He's the one that discovered if you boil milk, it doesn't kill you, right? People were dying, and he's like, what the heck? It's just milk. Also, whoever thought, like, I'm going to drink that right out of a cow, had some stones. What did he do? Someone tell me what Pasteur did. What is this diagram showing?
He did a series of tests with seeing if things really grew just straight out of whatever this broth was. He tried sterilizing it and then just leaving it open and stopping it and then making the curved tube and then seeing how something had to get in order to grow there, that it wasn't coming from the broth itself.
Yes. They had a limited understanding of bacteria in his time, and they said they just pop up out of nowhere, right? Bacteria just, poof, God put them there, right? And Pasteur was like, doesn't make a whole lot of sense. So he boiled some broth, and he had sterile broth. He knew it was clean, and then he left it out, and then it went bad. Okay? Happened all the time. Then he boiled some broth and sealed the flask, and it never went bad. And he said, some external force cannot get in. Then he boiled some broth in a swan neck flask, and no bacteria got in. And he said, okay, exterior bacteria climb into the broth and make it bad. With this swan neck, that U-turn, they can't make it, right? Had he left this out for like months, it eventually would have gone bad, right? He was doing this over the course of days. And then he broke off the neck, and boom, it went bad. And he said, okay, it is definitely some kind of external factor getting into the broth or whatever it is and spoiling it, okay? So Pasteur figured out the start of how microorganisms spoil things, like milk. What did Stanley Miller, Harold Urey, that's not one person. What did Stanley Miller and Harold Urey do? It's a very long name. It's a very white name, too.
They created an experiment where they simulated the early atmosphere, and in the end they were able to collect what they found complex biomolecules in the sample.
Yeah. So they put in what they thought would have been the chemical makeup of our earliest atmosphere, used an electrode to put some energy in there, right? And then they distilled it, condensed it, and got it down, and they said, there are some biomolecules in here, some RNA and things like that, right? And so they said, this may be an approximate simulation of how we were able to get life. These gases formed, some kind of electrical discharge like lightning, gave them energy to fuse together, and they formed RNA, which eventually formed DNA, which eventually formed living organisms, right? Now, it wasn't spot on. They didn't have it exactly right. They weren't able to create a new species out of this. They didn't Jurassic Park this crap. But it worked enough to show that this is kind of how life originated. Got it? Okay. So, was RNA the first genetic material? Question mark. We're not sure. Welcome to science. But we're pretty sure that it probably was. Welcome to science. They think that because RNA can self-assemble, right? DNA can't do that. Remember how we have to have so many topoisomerase and primers and helicase and all that, right? RNA doesn't need that. RNA will literally alphabet soup itself into words and build genetic codes just on its own, okay? So they said, well, if there's any genetic material out there that started it all, it's probably RNA, because it didn't need any help to do it. Okay? So I would just know RNA was probably the first genetic material. It's because it can self-assemble and because it can self-replicate rapidly. Okay? So it didn't take very long for these things to start going haywire and just start building living organisms, right? And RNA can make more RNA, right? Sort of how DNA can make more DNA, but RNA can essentially do it on its own with a little bit of like zinc and things like that, right? So RNA makes more RNA. You guys see how there's a pretty good amount of evidence pointing towards RNA being the first genetic material? It meets all the important criteria, so that's probably who started it all. Okay. Just a brief rundown of endosymbiosis. It's we've already learned it. It's your mitochondria and your chloroplasts that moved into eukaryotic cells for warmth and safety and love, right? And they give energy back to the cell, so it's a symbiotic relationship. It's endosymbiotic because they live inside of each other. Okay, which came first, mitochondria or chloroplast? Mitochondria. It's up there. It's up there in the picture, right? Plus the eyebrow. The eyebrow gave it away. Mitochondria. Okay. How do we know mitochondria came first? Besides the picture there. Huh?
Is it in both plant and animal cells?
It's in both, right? So had chloroplasts come first, plants and animals would have chloroplasts. Okay. Eukaryotic organisms got their mitochondria. Then we separated into plants and animals, and the plants picked up chloroplasts and the animals didn't. But we both had them to start with because we descended from each other, right? You guys see the logic there? Mitochondria had to come first in order for both of us to have them. All right. Domains, kingdoms, all that. This one is old school trash, right? Doesn't work anymore. We had our 5 kingdoms. I don't even know what Monera even is. They were like, this isn't a thing, right? This doesn't work. So they said, let's do 3 domains instead, right? Bacteria, archaea, eukarya. Which ones are prokaryotic? Bacteria and archaea. Eukaryotic? Eukarya, right? Now these each break down into their own kingdoms, but there's too many bacterial kingdoms and archaean kingdoms and protistan kingdoms to even keep track of, right? So we just say there's a lot of them, and then there's 3 more: plants, fungi, animals. Okay? We'll get into the specifics of each of them, but for now, just know the 3 domains: bacteria, archaea, eukarya, and then the 5 or 6 kingdoms. 3 of them are broken into their own separate kingdoms, but the 6 like head kingdoms: bacteria, archaea, protista, plant, animal, fungi. Okay? Be familiar with those 6. All right, these groups here on the bottom I can't draw. Okay, final slide. This is a what? It's a prokaryotic organism, okay? Memorize those 4 qualifications. What makes a prokaryote? A prokaryote has no nucleus. What does it have? A nucleoid region, okay? Few internal membranes, right? Pretty much just has like a plasma membrane and a cell wall. Doesn't have like an endoplasmic reticulum and a Golgi apparatus and nuclear envelopes and all that, right? It's pretty basic. No chromosomes, because its DNA is circular, right? So it doesn't make that sort of classic X, it's just a circle. And then it doesn't have, or it has a cell wall, but it's not made of cellulose, right? We all good on that?
Is it still made out of chromatin?
Huh?
Is it still made out of chromatin?
The bacterial DNA?
Yeah.
As far as I'm aware, yeah, it's made of the same material, but it's circular in shape. And it has obviously some differences.
Would it basically only have like 1 chromosome?
It doesn't have them, it just has a bunch of circles. And because they're circular, they're not considered chromosomes. They have to be at least close to that X shape to be a chromosome. It's a circle, so it can't have like a sister chromatid. It's a circle.
Are you talking like amino acid kind of circle?
No, the DNA is actually a circle, like it forms a loop. So our chromosomes form, it will not let me draw. It forms the X, right? You guys know what I'm talking about? Sister chromatid and sister chromatid makes a chromosome. You can't have a sister chromatid to a circle because they're circles, so they don't pair up side to side. I don't know exactly the mechanics behind it, but circular DNA does not form chromosomes. It's just considered circular DNA.
How does it replicate?
Huh?
These cells divide, right?
Yeah, and it replicates fairly similar to how we do, but it doesn't do like a bunch of replication bubbles. It does one replication bubble, and it goes in a whole circle. Cool beans? And we'll talk about this more on Wednesday, and Hillary will talk about it more on Tuesday. And we'll get into like gram positive, gram negative, all that stuff. We ran out of time. All right, thank you for coming.